Good morning, everyone. So, myself Abab, and today we are going to discuss the most important chapter of our human physiology, neural control and coordination, and the second we have chemical coordination and integration. So, these are the last chapter of human physiology from class 11. Now we'll discuss one by one in detail, and accordingly will solve some question related to these chapters. Okay. So the term coordination is very important for organs or any tissue. For it is the process. So it is responsible for day-to-day activities also. For example, if you are doing vigorous exercise or some physical exercise, there is a change in your internal body or homeostasis. Okay, so if you are running, your muscle will need more oxygen and more blood supply. That's why the heart rate will also increased breathing also. So all the organs inside our body must have coordination to different organs. Okay, so coordination is the process through which two or more organs interact and complement the functions of one another. For example, if organ 1 is situated in thoracic cavity and organ 2 is located on abdominal cavity, so for any physical activity or for any activity they must have communication between these two. That's why we have the neural system and chemical system. In neural system there is a nerve fibers present and which originates from the CNS, that means the brain and spinal cord. These these fibers innervates the organ or tissue and also organ too, so that they can communicate with each other. Okay, now for example, in neural system we have the brain and the spinal cord which is present inside the vertebral column of our body, and it is dorsally present. For endocrine system we have diffused tissues or organs. For example, the thyroid gland and thymus gland, adrenal gland, pancreas, gonads. So these two, neural coordination and the chemical coordination, combinedly coordinate and helpful for the any activity, any physical activity or day-to-day life activity. And these two systems must be in the synchronized way. Now the neural system. As we move from simple organism to the complex organism, the neural system is also becoming the more complex. For example, if we take the hydra, there are only network of neurons present. That means neurons are the excitable cells. Okay, they can receive and sense the stimulus and change accordingly and gives response to the organs or any tissue or any cell. So in hydra there is only network of these neurons, but if we talk about the insects, then brain with ganglia and thus this ladder like spinal cord is also present. Two ladder like parallelly at the ventral region present at the insects, inside the insects. So this is the brain and this is the ladder like neural tubes arising from the brain, and these are the ganglia part. which is present inside the insects. For example, cockroach. And in humans, we have most developed neural system, that is we have the brain and the spinal cord, which constitutes the central nervous system of our body, and all the nerves arises from this CNS are under the peripheral nervous system. Now we move to the next slide. So neural system, that is nervous system of our body, is composed of the highly specialized cell which we called neurons. Neurons are excitatory cells, and the function of neuron is to detect, receive, and respond according to the stimulus and transmit the different kind of stimuli also. And what is stimulus? Stimulus is the change in external or internal environment. So whether any change it is inside or outside of our body, it is stimulus which can sense by our receptors or sensory organs. We have 5 different type of sensory organs. They receive the stimulus and send the signals to the brain or CNS, central nervous system, and the CNS analyze it, and then the CNS gives command or respond according to their kind of stimulus. Same, in previous slide we have discussed the hydra have the network of neurons only, and neurons are the cell, but in insects number of ganglia are the collection of neurons, or we can say the cell bodies and neural tissues. In vertebrates, this is the well developed neural system, so that we can analyze each and everything accordingly. Now the human neural system. Human neural system consists of the central nervous system and the peripheral nervous system. In central nervous system we have brain and spinal cord. Brain is present inside the cranial cavity of the skull. Cranium of skull, bony structure, bony box, and in this inside this the CSF is present and the brain is suspended on this fluid. And then the spinal cord arises from this brain, which run across to the vertebral column, and the spinal cord vertebra, and all the nerves arises from this central nervous system are here in the peripheral nervous system. For example, afferent and efferent nerves fibers. Afferent are the nerve fibers from the sensory organs, and it transmits the impulse to the CNS. These are the afferent fibers. These are from the tissue or organ. Or we can say these are the sensory organs which receive the stimulus and gives signals or send impulse to the central nervous system. And efferent are the fibers which comes out from the central nervous system to the effector muscle. or tissue. So there is a basic difference that in afferent the signal will transmit from sensory organs to the CNS of our body, but afferent nerve fibers exits out from the central nervous system and to the effector or any muscle or any tissue. Now the type of peripheral nervous system. Peripheral nervous system is of two types, somatic and the autonomic. Somatic are the nerve fibers which transmit the impulse to the skeletal muscles, which gives commands to the skeletal muscles, or we can say these are the motor nerve fibers. And in autonomic, the CNS will give command and transmit the impulse or send the signals to the autonomic organs, or we can say visceral organs. For example, we have different kind of visceral organs: lungs, hearts, and kidney. So CNS, from CNS it gives impulse to the autonomic organs or visceral organs, or we can say smooth muscles of organs, and these are involuntary in nature. And but this somatic neural system, that means the signal from CNS to the skeletal muscles are involuntary. That means we can control by our own will, but these cannot be controlled by our own will. Now sympathetic and parasympathetic. Sympathetic are generally in the to reduce the emergency response, but parasympathetic are, for example, these two are antagonistic to each other. If there is a piloerection, that means it is the type of autonomic nervous system. We cannot control the piloerection, or any other. The dilation of pupil is an example of the ANS controlled by the CNS. Now visceral nervous system. So visceral organs sends the CNS in this. The sensory and motor, both nerve fibers are present, but ANS have only the motor neurons. So you do not have to confuse about the visceral and the autonomic nervous system. Visceral organs receive the signals from the CNS and also transmit to the CNS, but autonomic nervous system have only the motor nerve fibers. That means sensory and both motor. Sensory neuron or nerve fiber and motor nerve fiber both are present in the visceral nervous system. But in autonomic neural system, this is the motor neuron only. Now the question is, major part of peripheral nervous system is somatic neural system, which relays impulses from. The question is asking about the peripheral nervous system, that is the PNS, and somatic nervous system, which relays impulse from. And somatic means this is the skeletal muscles, and impulse is the transmission of signal. That means it originates after the sensing the stimulus and then it converts into the impulse. So skeletal muscles, and it arises from the CNS. So answer is this CNS2 skeletal muscles. The neural system is better organized in dash where brain is present along with a number of dash and dash. So in previous slides we have discussed that the hydra have a network of neurons. So this is the wrong statement. Now vertebrates, brain tissue and ganglia. Vertebrates had more complex nervous system. Rest, the insect have the ganglia and neural tissue respectively. So our right answer is the A option. The neural system is better organized in insects, where a brain is present along with a number of ganglia, that is the collection of cytons and the neural tissues. The function of our visceral organs is controlled by sympathetic and parasympathetic nervous system. Visceral organs, that means ANS also. In ANS we have sympathetic and parasympathetic, para and sympathetic, parasympathetic and that means the answer is sympathetic and parasympathetic neural system. Now the structure of neuron. Neuron is a is an excitable cell. That means its membrane is in polarized. Polarized. That, for example, if we talk about any cell, it has plasma membrane. This is the plasma membrane. If we cut this section from here to here and study, then it is composed of the phospholipid bilayers. And in cell biology chapter, you have studied that the cell membrane or plasma membrane also have transporters present. Some are active transporters, some are passive transporters. So because of this active and passive transporters present inside the across the plasma membrane, there is a gradient development. According to the ionic concentration inside the cell or outside the cell. And this is the outside cell, that is extracellular, and this is the inside, that is intracellular. So if we talk about the same, the neuron is also composed of the plasma membrane, and its plasma membrane also have the transporters which may be active and which may be passive. So these transporters present across this membrane and generate a gradient of the ions, gradient difference. And if we measure this voltage, then it comes out minus 70 millivolt. and here minus sign refers that inside negative or inner leaflet. So this one is the outer leaflet which face towards the extracellular, and this one is the inner leaflet, that is which face towards the cytosol or cytoplasm. And if we measure this potential difference by which is generated by these channels or transporters, so it comes out to be minus 70 millivolt. And here the minus sign is that the potential inside or across this inner leaflet is negative. So neurons are excitable cells, excitatory cells, because it can, the plasma membrane of this neuron can change its membrane potential or resting membrane potential. Each and every cell has some membrane potential. For example, epithelial or adipose also have some kind of membrane potential, but they do not consider in the excitable cells because they cannot alter their membrane potential. But if we talk about the neurons or muscle cells, they can change their membrane potential according to their transporters, or whether it is active or passive. In active transport, in active transport, this is the uphill process, that is from low concentration to high concentration, and energy is required, and it is against the concentration gradient. In passive transport, it is downhill from, in uphill process energy is required, it is from high to low, low to high, clear. In downhill, this is high to low, that is along the concentration gradient, and energy is not required. So passive transport are downhill process, and the flow of the ion or any particle is from high to low concentration, that is along the concentration gradient, and we do not require energy. And in our cells, the energy is in the form of ATPs. So in passive transport, no ATP is required, but in active transport, energy is required because it is against the concentration gradient from low to high concentration. That means ATP utilized. So the neuron is excitatory cells because it have the resting membrane potential, and it can change its membrane potential. It can reach up to the plus 30 or it can reach up to the minus 90 according to the opening and closing of the transporters or channels, ion channels. Now the structure of neuron. The structure of neuron have the cell body, this cell body part. Inside this, the cytoplasm, micelle granules are present, and along with the nucleus. And the next part of this neuron is that dendrites, branch structure, small branch structure, which receives the stimulus and then A long part is the axon. This one is the axon. This. And you can see that in axons, a specific cell is present, which is known as the Schwann cells. Schwann cells can be present in myelinated neurons also and non-myelinated neurons also. So Schwann cells are responsible. Schwann cells and oligodendrocytes are responsible for this concentric layers of the myelin sheath present around the axon. This pink color structure is the myelin sheath, and it is not continuous. You can see the gaps. You can see the gaps. This is not continuous. So these the absence of this myelin sheath is the node of Ranvier, and from axons, the last part, the ending part, the distal part is the axon terminal, which have the synaptic knob. This is the bulb-like structure, round swelling. So in neurons, we have the cell body or cytone part, and the second part is the dendrites, and the third part is the axon, which is the longest part of the neuron or nerve cell. Now the question is, select the correct option to represent A to E in the given structure of neuron. So here A are the dendrites, and B is the cell body or cytone, and C are the myelin sheath, and D is the axon, and E is the synaptic knob. So A dendrites, that means these two are incorrect. B is the cell body of cytone, and then C is the Schwann cell here, and D is the node of Ranvier. No, D is the node of Ranvier, and E is the synaptic knob. Yes, E is the synaptic knob here, and D is the node of Ranvier in both options. And now C, she is the Schwann cell. She is the axon part. This one is the axon part. Schwann cell are this, are this black color structure, Schwann cell. So our correct option is A. Now the types of neuron on the basis of dendrons and axons. We have three different types in our NCRT. First one is the unipolar, and the second is the bipolar, and the third one is the multipolar. So as we have discussed earlier, this is the multipolar structure in which there are many dendrites arise from this cytone region, many dendrites, and single axon. This is the longest part of neuron or nerve cell, and its example is in cerebral cortex. We have this multipolar type of neurons present, and then the bipolar, in which there is this one is the sensory part, and this is the dendrite, and this one is the axon. So in which bipolar have one axon and one dendrite arise from this cell body. This is the cell body, or cyton. So in by and the examples are the retina of our eye. And in unipolar, there is a single axon present inside the cell, and it is found in embryonic stage of our life. So this is the important slide. You have to memorize these things, that unipolar are in embryonic stage present in our body, but bipolar are present inside our retina of our eye, and multipolar, these are present in the inside the cerebral cortex region. Now, question related to the previous slide. Bipolar neurons are found in the embryonic stage. No, this is not the answer. Cerebral cortex? No. Cerebellum? No. Bipolar? So this is the right answer as we have discussed earlier. In bipolar, this the example is our retina of our eye. This can sense the light and receive and send it to the cell body, and then it will pass to other axons. It will pass to other dendrites or other neurons. Fibers which transmit impulse towards the cell body are called as. Now the question is asking about the fibers which transmit impulse towards the cell body. That means towards the cell body in this direction, not in this direction. Away from. This is the away from cell body. So answer is this branch structures. So branch structures are dendrites, not axon terminal, not axon. Axon which moves, the impulse moves away from cyton or cell body. Axon hillock is not the answer. Now there are some cells which are present in our neural tissue which are called as the glial cells. Glial cells are the oligodendrocyte cells, Schwann cells, microglial cells, ependymal cells. So Schwann cells and oligodendrocyte cells are responsible for these two, responsible for formation of myelin sheath. Formation of myelin sheath. Myelin sheath are the concentric rings of layers around axon. And this is not continuous, that's it is discontinuous. And where it is absent, the we can say there is node of Ranvier is present. Node of Ranvier is present. And Schwann cells and the oligodendrocytes, these two cells, both cells are responsible for the formation of myelin sheath. Myelin sheath are the concentric rings of layers which are present around the axon part, and it is discontinuous in nature, and where there is a absence of this myelin sheath, we can say this is the node of Ranvier. And now the topic is myelinated and non-myelinated. So myelinated neurons In myelinated neurons, Schwann cells is present. Schwann cells are present, and also myelin sheath. Along with myelin sheath, but in non-myelinated neurons, Schwann cells are present, but myelin sheath is absent. So this is the basic difference. In both myelinated and non-myelinated, the Schwann cells always present. But the myelin sheath is absent in the non-myelinated neurons. Non-myelinated examples are the somatic and autonomic nerve fibers, and myelinated we have the spinal and cranial nerves. The nerve arise from spinal cord, that is spinal nerves and cranial nerves. So in these nerves, the myelin sheath is also present along with the Schwann cells. But in this nerve fibers or nervous system, the somatic or autonomic, the myelin sheath is absent and Schwann cells are present. Now there are terms glia, there are terms cytone and nuclei and tract and nerve fiber. So if we talk about the CNS, the collection of cell body is known as the nuclei, and in PNS, the collection of cell body that is outside the CNS. This is termed as the ganglia, and collection of nerve fibers or axons. We can say this is the axon part. In CNS, this is known as the tract, and in PNS, we can use the term nerve. So nuclei are the collection of cytons, that is cell bodies, present in our CNS, that is brain or spinal cord. But if this collection of cell body present outside the CNS, that is in PNS, peripheral nervous system, we can use the term ganglia. Same with respect to the nerve fiber. So in CNS, if this nerve fiber present in the CNS or a collection of axons present inside the CNS, we can use the term tract, and in PNS we can use the term nerve. So our next question, myelin sheath is produced by, no, this is not the answer, not astrocytes, not osteoclast, not osteoclast. These are inside present inside the bone. So our right answer is the Schwann cells and oligodendrocytes. So Schwann cells and oligodendrocytes produce the myelin sheath. Generation and conduction of nerve impulse. So neurons are excitable cells. We have discussed this in earlier slides that neurons are excitable cells, excitatory cells because they can change their membrane potential from -70 to +30 and +30 to -90 and -70 also. So excitable cells, that is, can change their membrane potential, and it can receive stimulus. Stimulus is the change in external or internal environment, and it can transmit impulse and generate the impulse also. So it can transmit the impulse from one neuron to another neuron. And now the term is polarized. So neurons are excitable cells because their plasma membrane are in polarized state. So polarized means that means they have positive and negative. There is a gradient of ions, concentration gradient of ions. So let's see. So this is the neuron, and it also consists of plasma membrane, cytoplasm, and nucleus, basic cell structure. So if we cut the plasma membrane of this axon and study, then this is also composed of the phospholipid bilayer, and as we have discussed earlier that it have transporters or ion channels also. We use the term ion channels here. So there are different type of ion channels present inside the cell membrane. So let's discuss one by one. There are leaky channels, there are voltage gated ion channels, ligand gated ion channels, and also we have mechanical gated ion channel. But according to the NEET point of view, we just have to study only these two, leaky channels and voltage gated ion channels. In leaky channels, these are there are passive transport. That means same, it is a downhill process, and it is from high to low concentration along the concentration gradient, and energy is not required. And the examples of these leaky channels are potassium ion leaky channel or sodium ion leaky channel. So in NCRT, there is a line that in resting state, the plasma membrane of this neuron is more permeable to potassium ions. That means this line refers that the potassium ion leaky channels are more in number as compared to the sodium ion leaky channels. And now, so same, if this leaky channel is present, it will work according to the concentration gradient. It always move the potassium ions from high concentration to low concentration. And if we talk about the sodium ion leaky channel, then it is also moves or transports the sodium ion from high to low concentration. But if we talk about the voltage gated channels, these are open according to the stimulation. Opens only when stimulation is provided or stimulus is provided. And we have sodium ion voltage gated ion channel more than the potassium ion voltage gated ion channel, and it always open when there is change in voltage of plasma membrane. So these leaky channels are responsible for this generation of this membrane potential. So let us discuss how. And ligand gated ion channels examples are the calcium ion, LGIC, but you do not have to study or memorize these things. This is the phospholipid bilayer of a cell or neuron, and this is the extracellular environment, and this is the intracellular. Here we can say fluid is present, and intracellular there is cytoplasm or cytosol. As you have also studied, there are some proteins or protein part also present inside the cytoplasm. There is also central dogma, that means from DNA to RNA, RNA to protein. So proteins are always present inside this cell. There are some negatively charged proteins. And now there is a one pump which is also present in each and every cell, that is universal. The sodium potassium pump. Let's discuss this first. So sodium potassium pump, or we can say this is the ATPase. It is active transporter. First point is that it is active transporter. That means it utilizes energy in the form of ATP. And same, if this is active transporter, that means it always move ions or sodium or potassium from low to high concentration. This is against the concentration gradient, and sodium potassium pump always moves the or transports the 3 sodium outside and 2 potassium inside of the cell by utilizing the ATP, and it forms the ADP plus inorganic phosphate. So sodium potassium pump is present in the membrane of neuron. Now its properties are that it is an active transporter and it moves or transports the 3 sodium outside of the cell and 2 potassium inside of the cell by utilizing the ATP molecule, adenosine triphosphate. And in this plasma membrane, there are also leaky channels present. But in NCT, there is a line that in resting state, membrane is more permeable to potassium ion. So indirectly, it is saying that is potassium ion channels are potassium leaky channels are more in number as compared to the sodium leaky channels. That's why the gradient is developed by this potassium ion leaky channels and sodium ion leaky channels. These are the gradient developer. And the sodium ion concentration is always low inside the cell, and potassium ion concentration is always high inside the cell. And opposite, so leaky channels are responsible for this gradient development. The sodium ion is always in low concentration inside the cytosol or cytoplasm, but it is present in high amount in the extracellular fluid or outside the cell, because sodium potassium pump is responsible for the this pump is responsible for the maintaining the membrane potential. So this the leaky channels or ion channels are responsible for the generation of membrane potential according to their random open and close according to their concentration gradient. And sodium potassium ATPase is the one who is responsible for maintaining this membrane potentium. These are the gradient developer, and this is responsible for the maintenance of this. So now if you can see this, the positive part, there is 3 and there is 2. There is always difference of 1. That means one more positive always present inside outside the cell. So that's why the outer leaflet, this one is the outer leaflet like this. And this one is the inner leaflet of phospholipid bilayer. So this outer leaflet is always positive in resting phase, and the inner leaflet is always negative in resting state. This is the resting state. Now, if we measure, if we use the voltmeter for this measurement of this potential or potential difference between these two leaflets, outer and inner. So if we use this, then the voltage comes out to be minus 70 millivolt, and the minus is the inside the cell, and this is the RMP, resting membrane potential. So leaky channels or this potassium ion channels, leaky channels or sodium ion leaky channels are responsible for this generation of this membrane potential, and this pump is responsible for the maintaining the membrane potential. So you can see that there is always a difference of this one. There are 3 positive outside, 2 positive inside. There, that means there is more positive outside the cell. And always you have to memorize these things, that sodium is high outside the cell and potassium is high inside the cell. So that's why the neuron is excitatory cells, because it can alter, it can change from minus 70 to plus 30, and then plus 30 to minus 90, and again minus 70. So the membrane of this neuron can change their potential. That's why it is known as the excitatory cells. Neurons and muscles also are the excitatory cells, but epithelial adipose, these are not excitatory cells. Adipose tissue or adipose cell, they do not transmit the impulse, they do not generate this membrane, this plus 30 millivolt or minus 90, or they cannot change or alter their membrane potential. So same types of ion channels. First are the leaky channels, voltage gated and ion channels, and ligand and mechanical. So types of ion channels, we have leaky channels. For example, potassium ion leaky channels are more in plasma membrane of the neuron as compared to the sodium ion leaky channels. And now the voltage gated ion channels, the sodium ion voltage gated ion channels are more in number as we compare to the potassium ion voltage gated ion channels. So the leaky channels are always transport the ions passively. They do not require any energy. But the voltage gated ion channels always open when they when there is a change in voltage of the plasma membrane, and then accordingly they move and transport the ions, whether it is efflux or influx. Now in resting phase, the neuron is not conducting any impulse. Same, the plasma membrane is more permeable, to potassium ions, and the potential difference is of minus 70 millivolt. And who is responsible? Potassium ion leaky channels are responsible, and also sodium potassium ATPase pump maintains the membrane potential. As you can see in this slide, this is the sodium potassium ATPase or pump. It always move out the three sodium ions outside the cell and two potassium ion influx that is inside the cell, and the plasma membrane of this axon is in this phase, and this is the axon, this is the membrane, and this is the cytoplasm of this axon. Inside this there is a fluid that is cytosol or cytoplasm. So the outer leaflet is always positive, this one is always positive, you can see here, because of this leaky channels and the sodium potassium pump. That's why the development of this positive ions outside the cell, out in outer leaflet, and negative inside the cell. This is the inside the cell. This is the ultra structure of this plasma membrane. But this is the axon part. So now this is the positive, this is the positive because of the leaky channels. Because the leaky channels, potassium ion channels, and also sodium ion channels present on both sides. Now the depolarization phase. So whenever the neurons receive the stimulus or sense the stimulus, there are some receptors. For example, sensory organs receive the stimulus. So there are receptors present in our sensory organs. There are 5 sensory organs present inside our body which receive the stimulus. That means they can detect, sense the change in external environment or any internal environment. The receptor may be touch receptor, baroreceptor, or thermal receptor, any kind of receptor, light receptor. So when there is a stimulus, there are sodium ion VGIC present, and now this is the resting membrane potential. But when a neuron senses the stimulus, it starts opening the sodium ion VGIC, and then, as you can see, the sodium ion concentration is always high outside the cell. So when there is a stimulus, the sodium ion VGIC open, and there is influx of sodium ion, rapid influx. First, it will reach to the threshold potential. If they pass this threshold potential, then there is a surge, there is a gradual increase of this membrane potential. It will reach from minus 70 millivolt to plus 30 millivolt. This is the depolarization phase. That means when the more positive ions influx or enters inside the cell or neuron, this positive enters now. Now there is more positive. So that's why the polarity will get reversed. Now this positive changed into the negative if we compare to the internal environment of the cell. And this is the depolarization phase. So the polarity get reversed because of the rapid influx of sodium ions inside the cell, and the polarity get reversed. And it will reach from minus 70 to the plus 30 millivolt. So this is the depolarization phase. First, it will pass the threshold potential. If they do not cross this potential, then there is no generation of this action, this potential. Now, if there is this type of stimulus, then it will not reach to the depolarizing phase, or if there is any low stimulus. So it will direct it. So it is mandatory to reach up to minus 55 millivolt. This is the threshold potential. If the stimulus is not strong enough to cross this minus 55 millivolt, then it will not depolarize this membrane. And if the stimulus is strong enough to cross or pass this minus 55 millivolt, then there is a rapid influx. This is the shoot of overshoot of this phase, and it is considered as the depolarizing phase. And in this, the sodium ion VGICs are open. This green color line shows the depolarizing phase, and this is reached up to the plus 30. So in depolarizing stage, the action potential or the nerve impulse is generated. So it can pass from one neuron to from one place to another place. And now the next phase, you can see in this is given in your NCRT also. When there is a stimulus, and if we provide the stimulus to the neurons, the sodium ion VGIC opens, and there is influx of this sodium ions which reverse the polarity. Now the positive potential inside this cell or cytosol across the both of this membrane, and negative outside the cell. And now it transmits from positive to negative. It is the natural law. It transmits from this positive to negative, and here outside the direction of this inside the cell is from A to B. But if we talk about the outside the cell, the direction is from B to A, so that the positive will enters in our positive will move from this region to this region. So get the polarity of the membrane get reversed during the depolarization phase, and it will reach up to the minus 70 to plus 30, but they have to pass the minus 55 millivolt. So stimulus should be strong enough to cross this threshold potential. Now, the action potential term. So this is the term which is also used as a nerve impulse. So nerve impulse or action potential is the potential by which the nerve actually transmits the stimulus from one place to another place. You can see inside it is transmitting from A to B. So accordingly, it will get transmitted along the whole of the axon from the axon hillock to the nerve ending part. So this is the action potential or nerve impulse. Now the repolarization phase. So potassium ion VGIC also opens when there is change in voltage. So it starts from minus 70 millivolt, that is RMP, and now it will reach to the plus 30 millivolt, that is depolarization phase. So when the neuron membrane reach to this plus 30 millivolt, it triggers this sodium ion VGIC and it get opens. Sorry, the potassium ion VGIC. Sodium ion VGIC and potassium ion VGIC both are present on the axonal membrane or plasma membrane of the neuron. Now, when the potential reach to the plus 30 millivolt, it triggers the potassium ion VGIC and it gets open. So potassium ion VGIC, as we have discussed earlier, there is high concentration of potassium ion as we compare to the outside of the cell. Now, the potassium ions are high and low. So when it get open, so it moves from high concentration to low concentration. That means the positive charge efflux. You can see the positive charge efflux. So when there is plus 30, the positive charge moves outside of this cell. So it will reach from plus 30 to plus 20, then plus 10 plus 0, and then it will again move towards the negative potential. But these channels are slow. So you can see this is the repolarizing phase. In this, when we reach the plus 30 millivolt, the potassium ion VGIC opens, and there is outflux of, oh sorry, efflux of positively charged ions. So that's why it will first move from plus 30 to plus 20, then 0, and then again. But these channels are low channels, and now it will reach up to the more negative also. This more negative, and when they get closed, again from the activity of the sodium potassium pump, it will regain its minus 70 RMP. This is the resting membrane potential RMP. So they are very slow as compared to the sodium ion VJC. That's why they do not close when the potential reaches to the minus 70, and they remain open in this phase also. That's why we can see the hyperpolarization phase. This is the hyperpolarization phase. Because of this slow positive ion VGIC, voltage gated ion channels. This is the repolarizing phase. Plus 30 to minus 90. This is approx minus 85 or minus 90. So now plus 32, it can reach up to minus 85 millivolt or minus 90 millivolt. That's why we can see the hyperpolarization phase. Because of this slow sodium ion, slow potassium ion VGIC, and then again it will reach up to the minus 70 millivolt, that is RMP. So it starts from the minus 70 and always reach again to the minus 70. This is the depolarization when it reaches to the plus 30 millivolt, in which the action of sodium ion VGIC takes place, and when the potential reaches up to the plus 30, it will trigger or it will open the potassium ion VGIC, because it will only open when there is change in voltage. That's why the voltage gated ion channels, and now they are very slow, so that's why more and more positive outside the cell, more and more positive outside the cell. So we can see the hyperpolarization phase also of minus 90 to plus 85 millivolt, and then it again reaches up to the plus minus 70 millivolt because of the sodium potassium pump activity. Sodium potassium pump is universal and it is always open. And this graph, it starts from the minus 70, reaches up to the minus 55. This is the threshold potential. If there is low potential from the minus 55, or there is no strong stimulus, then the action potential cannot be generated. But when it's crossed this minus 55, there is the influx of sodium ion. Because of this sodium ion VGIC open, and this is the overshoot, and now it will reach to the plus 30, and plus 30 opens the potassium ion VGIC, because it is depend on the voltage change. When they sense the change in voltage, that is it reach to the plus 30, it get open, and they are very slow. They start influx of the potassium ions, and there is more and more positive, and remain open, and also cross this minus 70 millivolt RMP. So that's why more negative here. This is the more negative, and we can see the hyperpolarization phase, and then there is a sodium potassium pump which maintains this minus 70 millivolt. Now the saltatory conduction. Saltatory conduction is from nodes of Ranvier. This is the nodes of Ranvier. These are the nodes of nervine, and you can see the action potential jumps from nodes of nervine to nodes of runway, and it is very fast as we compare to the unmyelinated neuron. There is no myelin sheath, no myelin sheath. That means this is the same across all the axon. There is plasma membrane, and action potential run across this whole axon, from whole axon, from this axon hillock to the nerve ending, but it is in jumping manner, and less energy is required. In less energy required. So these are the saltatory conduction, and it is very fast. Now the transmission of nerve impulse, we can say there are synapses present between the two neurons. There is the axon terminal or synaptic knob present at the terminal part of the neuron, which there, and then it get connect, and there is a connection between these two. If this is a electrical kinase, there is a gap junction present between the two neurons. If there is a chemical synapse, there is a space present in which the neurotransmitter get released, and the neurotransmitter may be inhibitory or excitatory for the next neuron. So now the types of synapse: electrical synapse and chemical synapse. Electrical synapse and are very rare in our body. Very rare. Chemical synapse, there is a gap present between two neurons, and there is junction. There is no space present between the two neurons of this electrical synapse. There is no chemical required. There is no neurotransmitter required. You can see in this figure electrical synapse. The ions move from one axon of one neuron. This is the neuron 1, and this is the neuron 2. Now, this is the synaptic knob, and this is the dendrite of next neuron or neuron 2 or nerve cell 2. So, ions can cross this membrane easily through the gap junctions. Gap junctions only allow the ions to pass, and that's why they are very fast. There is no space present between these two. They are in the plasma membrane of both these two are in contact, and only the gap junctions are present which allow only the ions to pass, and then the impulse or the action potential transmit from one neuron to another neuron. This is the electrical synapse. And now if we talk about the chemical synapse, there is a space present between these two which is known as the synaptic cleft. And now the presynaptic knob plus this synaptic cleft and postsynaptic membrane. And there is this is the space, this plus this, both considered as the synapse. So this is the neuron 1, and this is the neuron 2, and there is a space present. This they are not in directly contact. There is no fusion of this plasma membrane of both of these two. So you can see there is a space present in which the neurotransmitter get released, and the neurotransmitter may be excitatory and may be inhibitory for next neuron or for the receptor. And you can see the nerve impulse transmit from the axon hillock to the this terminal end, the synaptic knob of this neuron. Now, the calcium ion plays very important role for the fusion of the so fusion of synaptic synaptic vesicles to the plasma membrane of synaptic knob. So calcium ion required for this vesicles fusion. That means without calcium ions, they cannot fuse to this membrane. And inside this vesicle, these are the vesicles. And inside this, the blue color particles, this these are the neurotransmitters. Neurotransmitters, and they packed inside the vesicles, and along the upon the action of calcium ions, it get fused to this membrane so that the neurotransmitter get released into the synaptic cleft, and then again you can see these are the receptors of the next neuron or post synaptic neuron, and this is the dendrite part of next neuron. So there are receptors present to receive the signals or to receive these chemicals or neurotransmitters of the previous neuron. So it may be excitatory or it may be inhibitory. Inhibitory, in which we have the example GABA, glycine, and there are many more examples. Excitatory, we have the acetylcholine, norepinephrine, noradrenaline, etc. So acetylcholine is always excitatory in nature. That means it excites the next neuron. And when there is GABA released or glycine, and they are in inhibitory nature. That means they inhibit the nerve impulse. They cannot pass. The nerve impulse cannot pass from this neuron 1 to neuron 2. So this is the chemical synapse. Now the question for the maintenance of ionic gradients across the resting membrane, that is resting, the cell is in resting phase, minus 70 million. Sodium potassium pump transport. This is the active transporter, and always free sodium out to potassium in 3:2 ratio. That means free sodium. These are incorrect. Outwards, right. Then 2 potassium out of the cell, 2 potassium into the cell, 3 out and 2 in. That means this is the right answer. And this is also incorrect. 2 potassium out. So the right answer is option A, 3 sodium ions out and 2 potassium inside the cell. Saltatory conduction means that the membrane potential changes along the entire length of axons. No. Only when there is an imbalance between the salts. No. In all or none fashion. Saltatory means jumping fashion. that is from one node of Renvir to second node of Renvir to third node of Renvir, and so on. So this is the right answer. Release of chemical messengers from synaptic vesicles is triggered by same as we discussed in previous slide. Calcium ion is responsible for the fusion of the synaptical vesicles to the membrane of the neuron. Now, the binding of neurotransmitter with the receptors open. The binding of neurotransmitter with the receptors open ion channels along the entry of ions, which can generate a new potential in the. Now, the question is asking about the neurotransmitter opens ion channels along the entry of ions, which can generate a new potential. So, it is asking about the which membrane, pre or post. So, binding of neurotransmitters, that means receptors are present. So, it is post synaptic membrane. Only receptors are present. So, the answer is post synaptic membrane. The binding of neurotransmitter with the receptors open ion channels along the entry of ions, which can generate a new potential in the post synaptic membrane. Transmission of impulse in neuron takes place from dendrites to axon. From dendrites, that means this small branch structure. These are the dendrites and these are the axons. So first, from here to here, it is receiving, and from this to this direction. So dendrites to axon, this is true. Now neurotransmitters are released at neuromuscular junctions. This is also true. Neurotransmitter, for example, acetylcholine released as the neuromuscular junction, as you have discussed, as you have studied in the locomotion and movement chapter also. So the correct answer is both statement are correct. Both statement 1 and both statement 2 are correct. Unidirectional transmission of nerve impulse. Now the direction is unidirectional. Transmission of nerve impulse through synapse fiber is due to nerve fiber is insulated by medullary sheath. This is sodium pump starts operating only at the cytone and then continues into the nerve fiber. Neurotransmitters are released by dendrites and not by axons. This is also wrong statement. Neurotransmitters are released by the axon endings. Neurotransmitters are released by the axon endings and not by dendrites. This is the right statement. Because neurotransmitters always released by these axon endings, that is the synaptic knobs. And in dendrites there are receptors present, not this neurotransmitter or any axon endings. So the correct answer is option D. Now the topic is brain. Brain is a part of CNS. In CNS, central nervous system, we have brain and the spinal cord. So brain is the command and control system of our body. It controls all the organs, whether it is visceral or whether it is skeletal muscles, cardiac muscles, or any intestinal muscle, or eye movement, hand movement, in locomotion and movement. It is also responsible for any physical activity or for any internal activity. There is a homeostasis also maintained by this brain. Homeostasis means, that means the constant internal environment, the constant temperature, constant pH of fluid or brain, constant pH of fluid or blood also maintained by the brain, by our brain. So it controls the voluntary movement, right? Balance of the body, that is posture, also functioning of vital involuntary organs, that is lungs, heart, kidneys, heart rate. We can heart rate. So from this, the autonomous nervous system, the heart rate can be increased, decreased. Kidney, if you drink more water, then they sense that you have more fluid inside the body, then command it will give command to the kidneys and do the appropriate actions. And in lungs, breathing rate can be altered. There are respiratory centers, cardiovascular centers, and for kidney there are hormones which is responsible for the reabsorption of some water and electrolytes which are. So now thermoregulation, that means the temperature of our body inside. The normal temperature of our body is the 98.6 degrees Celsius, or we can say 37, 98.6 degree Fahrenheit, or we can say 37 degrees Celsius. So it controls the thermoregulation and your hunger and thirst, because there is a structure present inside our brain, hypothalamus, which controls this center, hunger and thirst center, circadian rhythm, the sleep wake cycle of your body. Several endocrine glands. Endocrine means the ductless glands. They do not have ducts. They lack ducts. They pour their secretion directly into their bloodstream or lymph, and site of processing of vision, hearing, memory, intelligence. So intelligence, memory, all these are controlled by our brain. Now meninges of brain, that means they have layers. The outer one, and this one is the middle, and this one is the inner one. In between these two, the dura mater, and between the arachnoid, the space is present, which is known as the subdural space, and the space between arachnoid and pia mater is the subarachnoid space. And these spaces are not empty, or these spaces are filled with fluid. For example, the subarachnoid space is filled with the CSF, and CSF is the cerebrospinal fluid, which provide buoyancy to our brain, and it acts at the excretory site for the brain also. It excretes wasteful material the, into the CSF. The cells of the brain can excrete, or they can release the excretory component to the CSF fluid, cerebrospinal fluid. They provide nourishment or nutrition to the cells of the brain. And now, between these arachnoid and dura mater, subdural space, there is a fluid, serous fluid present. So dura mater is the outermost, and the pia mater is attached to brain. So you have to memorize the name of these meninges from outer to inner. Outer one is the dura mater, and middle one is the arachnoid, and inner one is the pia matter. You can also memorize the DAP for better understanding. Now the parts of brain. This is the forebrain. Forebrain consists majority of portion of the brain, major portion, and the reduced part and the very small region of brain present inside our cranium. It is the midbrain, and then it leads to the hindbrain part. In hindbrain we have cerebellum, pons, and medulla oblongata. In midbrain there are corpora quadrigemina, and forebrain have different lobes: frontal lobe, occipital lobe, parietal lobe, temporal lobe, and this is the major portion in which you can see the gyri and sulcus part, major and minor grooves. Also the outer part, outer region of the forebrain is the cortex, and the inner one has the and outer region is of gray matter. The inner one have the white matter. So we have two cerebral hemisphere separated by a tract of nerve fiber which is known as the corpus callosum. You can see this. This is the corpus callosum, the tract of nerve fibers, and there are two cerebral hemisphere present, and there is a groove present between these two hemispheres, and this part is the cerebrum. Cerebrum is the major portion of this forebrain, and now it also have the thalamus region, and this is the hypothalamus, that is the floor of this thalamus. Midbrain present centrally, and then we have pons, varoli, the cerebellum. This is the cerebellum, and this is the medulla. This is cerebellum, this is pons, and this one is the medulla, and they are the parts of hindbrain. In and the midbrain along with pons and medulla is consist of brainstem. Cerebellum is not included in the brainstem, and then the medulla oblongata leads to the spinal cord. Lobes, that means which is in front of our body, that is this in ventral region, the frontal lobe, and then we have parietal lobe, the occipital lobe, and the temporal lobe. Frontal lobe is responsible for the reasoning, intelligence, and also it can do mathematical equations very easily, and rest are the parietal lobe and occipital lobe and temporal lobe. Now you can see these are the two cerebral hemisphere from here to here, which is connected by a corpus callosum structure. This is the corpus callosum. Tract, a tract like fiber, a tract of fiber, and then this one is the left hemisphere, and this one is the right hemisphere. These are the gyri and sulci region. Also, sulci are also present, And these are the cerebellum structure which maintains our the body balance. And this is the brainstem. In brainstem we have midbrain with pons and medulla oblongata, and the pons varoli. So midbrain along with the pons varoli and medulla oblongata considered as the brain stem, which connects the brain to the spinal cord. Now the thalamus and the hypothalamus. So in hypothalamus there are centers which controls the temperature, body temperature, and urge for eating and drinking. That means there are thirst center present and hunger center present inside the hypothalamus, and it also have the neurosecretory cells. That means they secretes the hormones, also called hypothalamic hormones, because they are released by the hypothalamus also, hypothalamus itself. And now the hypothalamic hormones are GnRH, GHIH, GHRH, prolactin inhibitory hormone, CRH, TRH. So gonadotropin releasing hormone, growth hormone releasing hormone, prolactin inhibitory hormone, corticotropin releasing hormone, and thyroid releasing hormone, thyrotropin releasing hormone. That means these all are the hypothalamic hormones synthesized in the hypothalamus region. It also have the nuclei, for example, paraventricular nuclei or supraoptic nuclei. So these are responsible for the synthesis of oxytocin and ADH. Oxytocin and ADH. ADH is also known as the vasopressin, AVP. So hypothalamic hormones are the GnRH, GHIH, PH. These affect the growth, and these acts on the gonadotrope cells of the pituitary gland, prolactin inhibitory hormone, that is the milk production, and corticotropin releasing hormone, and this TRH, the CRH responsible for the ACTH release, TRH responsible for the TSH release. So these we'll study in the endocrine system. Now the limbic system. So inner parts of cerebral hemisphere along with some structures which is known as the amygdala and hippocampus consist the limbic lobe or made the limbic system, responsible for the emotional behavior also. So along with the hypothalamus, that means this, it can control our emotional balance also, and also the fear, excitement, rage, and motivation centers. So limbic system along with hypothalamus is responsible for these activities. Now the midbrain. Midbrain is characterized by this iter or aqueduct of Sylvius. This is the duct through which the CSF can pass, and in posterior lobe we have the four corpora quadrigeminal lobes, which are two in number and two in number, two upper and two lower region. So these corpora quadrigemina are responsible for the visual reflex and the sound reflex. So responsible for visual and sound reflex. They receive the visual and sound stimulus simultaneously and act accordingly. And this is the duct or cerebral aqueduct, which is responsible for the flow of CSF. And crura cerebri or crus cerebri or cerebral peduncle are responsible for the movement of limbs also. So this is the part of midbrain. It is very reduced part of the brain. Now the hindbrain, the cerebrum is highly convoluted. It have more and more gyri and sulcus, and there are also space present for the additional neurons. Now pons have the pneumotaxic center, which regulate the breathing. And in medulla, this is the medulla region. In medulla, we have cardiovascular center, respiratory rhythm centers, and gastric controls the gastric secretion of the. So in bones, they connect the thalamus or the hypothalamus region to the medulla, and there are also it interconnects the whole brain. And now the cerebellum function is to maintain or for the body balance. That is, if if you if anyone drinks the alcohol, then the alcohol will affect the cerebellum region, and then the person cannot walk properly because the cerebellum is responsible for the body balance or walking, and also it receives the sound sound signals. So pons and the medulla oblongata and the pons with medulla and midbrain, these are considered as the brain stem. That means the brain stem connects the brain part to the spinal cord. Same, the brain stem. Which of the following structure has very folded surface in order to provide additional space for many neurons? Highly convoluted. This is saying highly convoluted and additional space. The highly convoluted region is the cerebellum, and also cerebellum also have the highly convoluted space, highly convoluted folded or the region of this cerebrum. It contains many sulci and gyri. So that's why the answer is C. Now the question is hypothalamus is present at the base of the thalamus involved in following functions. Control body temperature, vomiting, secrete somatocrine, urge of eating and drinking. So now it is saying except. So hypothalamus function is this control body temperature. That means this is the thermoregulation, right? secretes somatocrine, yes. The urge of eating and drinking, right? But this is not controlled by the hypothalamus. A canal passes through the midbrain is called cerebral aqueduct, through which the CSF can be can flow. Which part of brain is involved in loss of control when a person drinks alcohol? Where is the question? Cerebellum, because it maintains the body balance and through which a person can walk easily. Respiration, respiration, heartbeat, and peristalsis are controlled by. Now the respiration, heartbeat, and peristalsis. That means the gastric movement controlled by the medulla oblongata. Because medulla oblongata have respiratory centers, cardiovascular centers, and gastric control. Human hindbrain comprises three parts, one of which is hindbrain have the midbrain and then the midbrain, the hindbrain. The human hindbrain comprises three parts of which. So cerebellum plus bones plus medulla. So this from these three, no hypothalamus, no, no. So this is the right answer. Which of the following option correctly include parts of limbic system? So hippocampus, amygdala along with the hypothalamus. Cerebellum of brain is maintenance of posture and equilibrium, olfactory function, optic option, body balance. So cerebellum is responsible for the maintenance of posture and also body balance. If a person drinks the alcohol, then the cerebellum part of the brain will affect and then the person cannot walk properly. The parts of human brain that helps in regulation of sexual behavior, expression, expansion. This is the limbic system and hypothalamus along with hypothalamus. This is not corpus callosum, no, no brain stem or epithalamus. An area in brain which is associated with strong emotions is always the emotional center, the limbic system. Which part of brain is responsible for thermoregulation? Very easy question, hypothalamus. Corpus callosum connects or joins the two hemisphere. Medulla oblongata have the respiratory center, cardiovascular reflexes, and the gastric stimulations. But cerebrum, different functions. But here the question is asking about the thermoregulation. That's why the answer is hypothalamus. Select the incorrect statement regarding synapse. So select the incorrect statement regarding synapse. This is the EYQ from 2022. Impulse transmission across a chemical synapse is always faster than that across an electrical synapse. Always faster. Mhm. This is the right statement, but this is the incorrect statement. So the correct answer is option A. Missile body are mainly composed of the pre. These are actually the ribosomes and the RER, not protein, not lipid, not DNA, not nucleic acid. So rough endoplasmic reticulum along with ribosomes are the Nissel's bodies, which are present inside the cytoplasm of the cell body and the dendrites. Myelin sheath is produced by variegation. The oligodendrocytes and the Schwann cells receptor sites for neurotransmitter are present on the receptors, always present on the post synaptic membrane. So this is the right answer. A diagram showing axon terminal and the synapse is given. Identify correctly at least two of A to D. So C neurotransmitter, no. Synaptical vesicles, C is the synaptical vesicles. A is the neurotransmitter, B is the synaptic cleft, B synaptic connection. Neurotransmitter D is C is the neurotransmitter and D is the calcium ion. No. So A is the neurotransmitter here, which is present between the space of these two neurons, and B is the synaptic cleft. A is the receptor, C is the synaptical vesicles. A is the receptor and C is the synaptical vesicles. So this is the right answer. Here B is the synaptic cleft and these are the vesicles, receptor. Now next question. Match the following. Now the hypothalamus have the neurosecretory cells and the medulla controls the respiration. Pons connects different region of the brain. And cerebellum have the additional space for neurons. So the right match is A to third and B to fourth and C to second and D to first. So do not think about the options, just match list 1 and list 2. The answer will come out very easily. The cerebral hemispheres are connected by nerve tract known as corpus callosum. Right, the brainstem consists of the medulla, pons and cerebellum, not cerebrum, cerebellum. So this statement is wrong. So statement 1 is right, correct, and statement 2 is incorrect. That means the first option is the right answer. Now the parts of human brain that helps in regulation of sexual behavior, expression or expression of excitement, fear, pleasure, rage, etc. are limbic system. There is no corpus. This along with the hypothalamus. Brainstem of human brainstem of human brain consists of three regions. First, you have to see the midbrain, and there is no option other than A which have midbrain. So our answer is A. But just you have to see the midbrain pons. After seeing the midbrain, you can easily tick option A. Identify the region of brain which has pneumotaxic center. Pneumotaxic center, that means pons, and alters respiratory rate, reducing the rate. This is the pons region which has the pneumotaxic center, which alters the respiratory rate and reduce the rate of inspiration, which I have discussed, which you have studied in the body fluids, in the respiratory system chapter. Responsible for thermoregulation, hypothalamus, no cerebrum, no corpus callosum, no medulla. Which of the following structures or regions is incorrectly paired with its function? Medulla oblongata controls respiration and cardiovascular reflexes. Limbic system consists of fiber tracts that interconnect different regions of the brain. No, limbic system does not connect the different regions of the brain. So, question is asking about the incorrectly paired. So, this is our right answer. Production and release. This also right. This is also correct. This is also correct. Injury localized to hypothalamus would most likely disrupt body temperature because it is responsible for the thermoregulation. Short-term memory, no. Coordination during locomotion, no. Executive functions such as decision making, no. Because hypothalamus is responsible for the thermoregulation. It have thirst center, hunger center. So that's why the correct answer is the regulation of body temperature. Now our next chapter is the chemical coordination and integration. So in this chapter we'll study basically about the endocrine system or hormones. For the in neural chapter we have studied that there is point to point connection between the organs. That means nerve fibers are connect. Nerve fibers are the way through which they can communicate, or neurons. But there is no point to point, that there is no innervation of nerve fiber to each and every cell. That's why we require this chemical coordination and integration system. That means the endocrine system. So basically it is responsible for the morphological changes or the any physiological change for the specific tissue or any cell, because hormones can act on each and every cell because of the target cells, and target cells have specific receptor by which they can bind to their specific hormones. Same, the neural system is point to point, and this is very fast. But if we talk about the physiological changes, we require the hormones. For example, steroid hormones, testosterone and estrogen and progesterone. They are responsible for the secondary sexual character. There is a permanent change in the physiology of a person. Now, neural system with endocrine system or the chemical coordination. maintains the homeostasis of our body. This is the internal environment which remain constant, which have to be remain constant. Otherwise, the body physiological activities alters. So hormones, there is a definition in NCRT. Hormones are non-nutrient intercellular messengers and which are released in very trace amount and have the physiological changes, and endocrine means inner away, and they do not have ducts. Endocrine glands do not have duct. Exocrine glands have duct. Endocrine glands are ductless glands. That means they pour their secretion directly into the bloodstream. There is no duct present for the secretions. Same, the secretions or the chemicals released by these glands or systems called as the hormones, which directly enters into the blood or bloodstream. For example, this is the endocrine cells. They synthesize some chemical or hormones, then they directly diffuse or directly secrete their chemicals into the bloodstream. This is the blood vessel. Blood is flowing, and so the circulation or circulatory system, because heart is pumping the blood 72 times per minute, so they can reach the target cell, because target cell have receptors. So this is specific. Each receptor has a specific. Each receptor is specific for a specific type of hormone. Now, if we talk about this and exocrine gland, these are the cells which secrete their chemicals. There may be secretions which may be water or enzyme. And now this is the duct. This is the duct through which it will travel or pass, and releases. So this is the duct, and they do not have duct, ductless, and they have duct. Duct is present. Hormones. The This is the very important definition. Non-nutrient. That means they do not provide energy. Just like if we eat the carbohydrate, protein, or fat, they provide energy into our body. But these hormones, if we take or if we ingest the hormones, they do not provide any nutrient or any energy. Intercellular means between two cells. If we talk about the pituitary gland, it will release hormone into the bloodstream and act. If there is a TSH, then it will act on the thyroid gland. So the pituitary gland cell and the thyroid gland cell are two different cells. That's why this is between two cells and messenger. It has some message. It will give command or alter the physiology of the respective cell or target cell. That's why it acts as a messenger, and it is very trace amount, produced in very trace amount. That means very low concentration but highly potent. They can change the physiology of any cell according to the type of hormone. They maintain the body homeostasis. Now, what are tropic hormones? For example, ACTH. ACTH is released by the pituitary gland, but it acts on the adrenal cortex, and it will also release hormones. So those hormones which are responsible for stimulation of this other gland and for the release of other hormones from the different part of body known as the tropic hormones. So question, hormones are, where is the question? Nutrient chemicals? No, because these are non-nutrient. Intra, here the term, you do not confuse. This is inter, okay, not intra. And produced in high amount? No, no high amount. Chemical messengers? Yes. So hormones are non-nutrient, intercellular, and produced in very trace amount or very low amount. And intercellular messengers, chemical messengers. Hormones and their classification. The hormone word coined by the Starling. Starling was a scientist. Now they may be inhibitory or they may be excitatory or stimulatory. And hormone is, the meaning of hormone is to excite, and secretin was the first hormone detected. Most of the hormones have the target cells or target sites, but two hormones, that is the TSH and the GH, sorry, and the thyroxine. The GH and the thyroxine acts on nearly the all somatic cells for the physiological changes, but TSH will only act on the thyroid gland. That means the receptors will always present at the thyroid gland. ACTH will always act on the adrenal cortex. So they are highly specific, but thyroxine and growth hormone targets all the cells of the body. And there are some disorders when any hormone release in hyper or hypo condition, then it will leads to the disorder or deformity. Now hormones are short-lived and they excreted out by the kidney also. First it will degraded by the liver, and in vertebrates there are many hormones which alter the physiology. For example, after reaching the age of puberty, the GnRH will release for the first time from the hypothalamus. And we are also vertebrates, but higher invertebrates have very simple endocrine system, but and they also produce few hormones. And if we compare ourselves to the invertebrates, then our endocrine system is more developed, and there are many hormones which are released daily. Endocrine glands are present at the in the diffuse tissue or cells located in the different parts of our body, human body. For example, We have pituitary gland situated or located at the brain inside our brain region attached to the forebrain. Pineal gland also inside our brain. And if we talk about the thyroid gland, it is present at the neck region and attached to the trachea. Pineal gland, ventral region, and it is present, situated or present at the sternum, thymus gland, and then pancreas, adrenal gland. So they have different locations. Pancreas, adrenal gland present at the inside the abdominal cavity, but thymus is in thoracic cavity. So this is the diagram from your NCRT. This is the hypothalamus, which is also a part of forebrain. In diencephalon part, we have the thalamus, epithalamus, and hypothalamus. Hypothalamus is the floor of the diencephalon. Now this pineal body, which is also present inside our brain, pituitary also inside our brain. This is the pituitary, which is connected to the hypothalamus through a stalk-like structure. Now the thyroid and parathyroid present at the neck region, attached to the trachea of our body, and then the thymus, which is attached to the ventral, attached to the flat bone, that is sternum, and present at the dorsal side, sorry, ventral side of our aorta. Now pancreas, and in the abdominal cavity, adrenal gland also. It is also known as the suprarenal gland. It have two different regions, cortex and medulla. And in males, their gonads are present, testis, extra abdominal cavity, form which is which starts the process of formation of sperm and testosterone after reaching the puberty. In ovaries, there is secretion. Ovaries, they secrete the steroid hormones, estrogen, progesterone, and these both gonads are responsible for the development of secondary sexual characters in the respective gender. Now this is the hypothalamus. Hypothalamus is a part of thermoregulation also, hunger center, neurosecretory cells, nuclei are also present, nuclei. That means it is the collection of cytons which synthesize the hormones called as the hypothalamic hormones. For example, GnRH, GHIH, GHRH, TRH, TRH, and PLIH. That means prolactin inhibitory hormone. These are the hypothalamic hormones synthesized in the hypothalamus region. Now it is connected to the pituitary through a stalk-like structure which is called as the infundibulum. And then the posterior pituitary is in the direct neural regulation of the hypothalamus, and the anterior pituitary is in the portal circulation. Is connect. There is a coordination or between these, the anterior pituitary and the hypothalamus through a blood flow. That is the portal system, hypothalamo-hypophyseal portal system. So, if we talk about the nuclei, there are two nuclei present, paraventricular. There are many more, but if we talk about the paraventricular and the supraoptic, they release the ADH and oxytocin, and they also produce hormone. ADH and oxytocin produced in the hypothalamus region. You have to memorize this, and it get transported along the axon part of these neurons, and they have terminal endings or the nerve endings in the posterior pituitary. So whenever there is a need, the posterior pituitary release the oxytocin and ADH. Nuclei, or we can say neurohormones, wide range of body functions. Same. Now This is the hypothalamus region, and we have a connection between the pituitary gland and the hypothalamus, which is known as the infundibulum here. Infundibulum is a stalk-like structure present between the hypothalamus and the pituitary, anterior and posterior. Now, this infundibulum, along with the pars nervosa, which is known as the posterior pituitary, the infundibulum, along with the pars nervosa, called as the neurohypophysis. And now, this is the pars distalis, that is anterior pituitary, and this is the pars tubralis, and there is a fused region of the mi- middle lobe of pituitary, which is known as the pars intermedia. So, in humans, the pars intermedia is fused with the pars distalis region, that is the anterior pituitary. It is fused. So, pars tubralis, pars distalis, and pars intermedia collectively called as the adenohypophysis. That is this plus this plus this. And now the infundibulum and the pars nervosa, collectively called as the neurohypophysis, and both adenohypophysis along with the neurohypophysis called as hypophysis cerebri, or we can say pituitary body. This is the pituitary body or pituitary gland. So now, if we talk about the hypothalamic neurons, that is this, its axon. Axon will end or terminates at the posterior pituitary region, and there is a long axon which run across the infundibulum. But for anterior pituitary regulation, there are neurons which just have the nerve endings at this region, at the infundibulum region or before the infundibulum region. So the posterior pituitary hormones are actually synthesized on this region, and then it get travel along the axon and reach the posterior lobe of pituitary. For example, ADH and oxytocin, they synthesized here, produced here, and then it get transport along this infundibulum region through the axon part, and then it get stored in the posterior lobe of pituitary. But if we talk about the anterior pituitary regulation, the hypothalamic hormones released at this site, and then there is a portal circulation, just like this. These are the blood vessels, that means arteries, veins, and capillaries. This is the capillary network. So this is the hypophyseal. This is the hypophyseal portal system. This hypothalamo hypophyseal portal circulation of system. These are the capillary network, and this is also a capillary network. So hormone are synthesized in this region, and then from the axon it can travel, and then it will released in this region, and through the blood circulation it get enter into the capillary network, and then through the circulation, because blood is always flowing, it will reach to the pituitary cells, which is also known as the pituocyte. There are the cells. These are the pituocytes. Act on these pituocytes. They act on these pituocytes, and then these pituitary cells release again these hormones into this capillary network, and then it gets released by the pituitary, or it enters into the systemic circulation. This is the systemic circulation. That means whole body circulation. And if we talk about this posterior lobe of pituitary, how the hormones enter into the systemic circulation. So this is the pathway of this circulation. So first, the hypothalamic hormones synthesized in the nuclei, and then it get travel along the axon, just like this axon, and then it will released into the bloodstream, and it may store in the posterior pituitary cells also. So from this they enters into the systemic circulation. So posterior lobe of pituitary stores and releases the ADH, or we can say vasopressin and oxytocin. Middle lobe of pituitary releases the MSH, that is melanocyte stimulating hormone, and anterior lobe of pituitary releases the six type of hormones, which is known as the GH, LH, FSH, TSH, ACTH, and prolactin, that is PL, released by the adenohypophysis only. So from adenohypophysis or anterior pituitary, that is pars distalis, there are six type of hormones they secrete, and from middle lobe of pituitary, the MSH will release melanocyte stimulating hormone, will act on the melanocyte that is responsible for the pigmentation of skin. And now the ADH and vasopressin and oxytocin synthesized or produced by the hypothalamus itself, and then travel along the axon and reach to the bloodstream from the posterior pituitary region or pars nervosa. So this is the hypothalamus connection with the pituitary gland. GnRH is produced by, this is the hypothalamic hormones, that's why this is, the answer is hypothalamus. Somatostatin produced by hypothalamus. So somatostatin it affects the growth. That means stimulates the release of growth hormone, inhibits the release of growth hormone. There are two, somatokinin and somatostatin. Statin. Growth hormone releasing hormone is responsible for the release of growth hormone, but somatostatin is responsible for the release of inhibition of the release of growth hormone. Pituitary gland is located in sella turcica. There is a bony cavity in sphenoid bone. Butterfly shaped bone present. H shaped bone. Pars intermedia, that is the middle lobe, is almost merged with the anterior lobe of pituitary, that is pars distalis. Pars nervosa is the posterior lobe. Pars tubralis is also a part of adenohypophysis. Posterior pituitary is different. Neurohypophysis through. Oxytocin and vasopressin are transported to neurohypophysis through long axon. There is no blood supply, there is no fluid, there is no lymph, only axon. Now this is the pituitary and this is the sphenoid bone, and this cavity, now you can see the depression at the bone, inside the bone, is known as the sella turcica. And this is the hypothalamus region, and this is the infundibulum, stalk-like structure present, and this is the it has circulation. This is in direct neural regulation, that is this is posterior pituitary, this is posterior pituitary, and this is anterior pituitary, this is infundibulum, this is hypothalamus. These are the neurons which travel along this infundibulum and reach to the posterior lobe of pituitary. And this is the circulation, that is hypothalamo-hypophyseal portal system. Same, pituitary is also known as the hypophysis, and it is small, round body, have direct connection with the hypothalamus, which is also known as the stalk or the infundibulum part. This is the sella turcica cavity and sphenoid bone. This is the bone region, and this is the sphenoid bone, butterfly shaped and H shaped. Now this is the posterior pituitary. You can see the axons are travel along the whole infundibulum and reach up to the posterior pituitary. And this neurons terminates here, and then there is a circulation portal system, hypothalamo hypophyseal portal system. So the hormones synthesized in this region travel along the axon and get stored or may release. Hormones synthesized in this region secreted at the stalk, and then through circulation it will travel and then act on the pituitary cells, and pituitary cells release their respective hormones, and then to the whole body circulation. Posterior, we have oxytocin, ADH from the posterior pituitary, and from anterior pituitary we have gonadotropins, that is FSH and LH, growth hormone, prolactin, adenocorticotropin hormone, thyroid stimulating hormone. So from anterior pituitary we have 6 different type of hormones. From middle lobe of pituitary we have 1, and from posterior lobe of pituitary we have 2 hormones. So total 6 plus 2 plus 1, that is 9 hormones released by the pituitary gland. Same, six from anterior pituitary, one from middle lobe of pituitary, pars intermedia, which is fused with the anterior pituitary in case of humans. Stop.